second question here. So here they are showing types of polysaccharides. So when you have this, when you are told that this is polysaccharide, usually it relates to sugar as the smallest unit. So here you have your basic unit of this. This would be your glucose. And then one very important point from this paper that would be useful to remember is that if you have this shape, this is called like a branch shape. And if you have this multiple branch, so this is known as highly branched shape, and this is a linear because it's like this. So this is the linear shape. So remember, for branch shape, and anyhow they are asking you what this is. So an example of branch shape polysaccharide would be the starch. An example of highly branched polysaccharide would be glycogen, and a linear example would be cellulose. So branch is starch, highly branched is glycogen, linear cellulose. Okay, and then the second part, what is the basic unit for all of them? So this we have already shown. The basic unit is this one here, and this is of course your glucose. And then X, which is starch, is a polymer that is made up of hundreds of monosaccharides, and you are required to explain how X can be broken down into monosaccharides. So monosaccharide in this case is the glucose. So you first can say what is the process. So the process to break down X is known as the enzymatic hydrolysis reaction. So that will be your point number 1. And the second point would be to name the enzyme. So amylase would be one very good example to state here that enzyme amylase can be used to break down this starch. Moving on, here they show you a diagram where you have a glucose plus fructose, and then you have a molecule P and water. So first of all, usually this is how I would attempt the question. So first of all, try to, because if they have X and Y and P, it means you have to identify them, or at least know what they are at a certain point of time. So molecule P, remember glucose and fructose, you will always get sucrose, because sucrose is spelled as S-U-C-R-O-S-E. So remember this R here and the R in fructose. These are the one way for you to remember. So glucose plus fructose is sucrose. Alright, so here the P is sucrose. You have already answered this part here. So what is the process here and here? So the process that produces water at the end of it will be known as the condensation process, and the process that uses up the water and breaks down the product using water will be hydrolysis. So remember, the process that produces a water molecule here is easier to remember it as condensation, and if you use the water to break down your molecule, this will be known as hydrolysis. And then moving on to another diagram here, 2.3 is talking about the transport of nutrients and assimilation by the liver and body cells. So you are required to explain what is this assimilation of glucose. So again, remember, whenever you are asked to explain something, usually you will be also given at least a diagram, and always use the keywords in the diagram given, and always try to look at the diagram to get some idea how are you gonna explain the process. So over here in this diagram, you have a few keywords that you can use. So you have a liver, you have glucose, lipids, glycerol, body cell. I think this should be enough. So step 1, you can say from the beginning. So step 1 would be excess glucose in the blood is converted to glycogen and stored in the liver. Okay, so this is your step 1 here. And then when the glucose level in the blood is low, remember, glycogen is then converted back to glucose. And then next, the liver is also able to convert excess glycogen into lipids. And then afterwards, upon reaching the body cells, which is this one here, the energy is released in cellular respiration by the oxidation of glucose process. So basically what you are trying to say here, if you want to remember it, would be first how glucose is converted to glycogen and where it's stored. So glucose is converted to glycogen stored in liver. Next, when the glucose level is low, you have the reverse process, right? So the glycogen is converted back to glucose. So glucose to glycogen in liver, glycogen back to glucose. And then if you have more glycogen, this can be changed into further stored into lipids. And the energy is released when you oxidize glucose. So this will be the 4 points you can use to explain this diagram.